Thank you for the introduction. My boss was supposed to speak today, Steve Petru, but he couldn't make it, so I'm standing in for him. Steve used to be the director of the Florey Institute of Neuroscience and Mental Health in Melbourne, Australia, and he's also the co-founder and chief scientific officer of Praxis Precision Medicines. I think most of you would have heard of those. We have several collaborations with Praxis to develop ASOs for neurodevelopmental diseases at the Florey, and I lead those for Steve. So recently we've got some philanthropic funding for SLC6A1 disease. We're trying to develop some ASOs and get some commercial interest in those to take that program further. And because of that, I won't be able to speak very much today about exactly how we're doing this and what the modes of action of those ASOs are until we've got that IP nailed down. That being said, I don't know how to use this. I'm pressing all the buttons and nothing's happening, so. Still nothing. Okay. All right, so the molecular mechanism of SLC6A1 haploinsufficiency. So when there's not enough of the protein made, SLC6A1 codes for a protein called GAT1, which transports GABA across cell membranes, in addition to sodium and chloride ions. The idea is that when one allele, one copy of SLC6A1 doesn't work properly, there's not enough GAT1 protein to sufficiently maintain the balance of GABA across the cell membrane. And the idea is that you get too much GABA hanging around and it potentiates the action of GABA receptors on postsynaptic membranes, and GABA's not doing its job properly. And its job is to dampen excitability of neurons in the central nervous system. So our idea is that what we can do to counter that. Is there somewhere I'm supposed to aim this? Okay, so the idea... Pardon? Aim it towards the back. I'll try that. Shield your eyes. The idea is that SLC6A1 gene gives rise to different variations and species of SLC6A1 mRNA, and that mRNA is used as a message to make GAT1 protein. And there are a number of ways that we can use small synthetic pieces of genetic material that are chemically modified to target regulatory sequences on those mRNA species and essentially remove the brakes from GAT1 protein expression. So effectively what we're doing is increasing the level of GAT1 protein and trying to overcome the phenotype of the disease using that mechanism. This is where it gets into the area where I can't say exactly what mechanisms we're using and what the targets are with those ASOs to increase production, but hopefully in the very near future we'll be able to elaborate a bit further on that. Work that time. So this is the sort of the pipeline that we've developed at the Florey, and we're in, now it just works all the time. So we're in the white squares there. At the moment we're screening in immortalised cell lines, ASOs, to see if they can upregulate SLC6A1 and GAT1 protein, and we're refining the design of those little pieces of genetic material to make them safer and make them work better. Ultimately what we want to end up with at the end of the pipeline is 3 to 5 potent, safe and durable ASOs that we can take into either a little bit further development for N-of-1 trials or into non-human primates for GLP tox, and then we can start to build a case for an IND application with the FDA. So, like I say, we're at the in vitro screening stage at the moment. There's some results that we've gotten in the last few days that I haven't been able to prepare. We just arrived today, so we're a little bit behind the 8 ball, but I can show you some of the preliminary screening results. This is in immortalised cells, so we've developed assays in those cell lines, and we've done some work to validate the mode that those ASOs are working through and the chemistry of the ASOs. And these are some preliminary screening results. So what we're seeing is at the mRNA level, we're getting up to 1.5 times as much SLC6A1 mRNA. I don't have the data to show you today for protein, but we have validated some of the best ones at the protein level with Western blot and LCMS, and typically we get a better upregulation at the protein level, and that's probably because the mRNA gets turned over more quickly than the protein. So the protein accumulates in the membrane. It's getting turned over every couple of days, where the mRNA is continuously turned over. So, you know, the most simple way to put it is that anything that's a twofold upregulation of the protein, so 200% as much of the protein, should overcome haploinsufficiency because you've got normal levels of GAT1. So we're pretty happy with those results. We do have some more in vitro screening to do, and we also have in vitro tox assays. And early in the new year, probably our best lead 10 to 15 candidates will go into animals for the first round of in vivo studies. Thank you. Do you want the old one?
Sure.
Thanks. Nope. Now it just must be me. Somehow I just blocked the signal. Okay. So, the first ones to go into in vivo studies will go into a mouse model that we specified about a year ago and has already been made now. And we have that, we're breeding it up for trials in the lab. So Melody Lee, sitting over there, is the head of our in vivo physiology team, and Mel will be responsible for these experiments. But what we have is a mouse that expresses the full-length human version of SLC6A1. So it's got the untranslated regions at the front and the back of the gene, and all of the introns and all of the regulatory elements of human SLC6A1. And we've also put a tag on the protein, so it makes human SLC6A1 protein with a tag on there that we can easily recognise with an antibody. And that's just to allow us to easily track the protein levels in the mouse cerebral cortex and spinal cord. So those mice, the first round of screens that we do is 14-week efficacy and safety. So we look for inflammatory markers in the central nervous system. We also do functional observational battery over those 14 days to make sure that it's not causing behavioural problems. And then at the end of the 14 days, we assess for efficacy at the protein and mRNA level. How much were we able to upregulate human SLC6A1? Once we do a bit more structure-activity relationship development of those ASOs, they go into 16-week trials in those same mice, and we're looking for the same things. We're looking for potency, safety and duration of action of the ASOs. So over the 16 weeks we do PD, PK, we measure levels in the brain and spinal cord of protein and mRNA. We look for inflammatory markers at different time points during that time, and that's essentially how we choose our final lead candidates. Okay. In parallel, we want to know, can we reverse the phenotype of the disease by increasing the levels of GAT1? It seems like that's a no-brainer, but is it possible to do that? And at what points in development can we do it? And how much do we need to do it to overcome the phenotype of the disease? So to answer those questions, we have another mouse model in which one of the SLC6A1 alleles has been deleted. So it's the haploinsufficiency model. And using tricks with CRISPR, we can upregulate the expression of that remaining allele to different amounts and at different points during development. And the idea of those experiments is to see, can we do it? Do we need to do it early in development? Can we do it at different points in development? And how much do we need to upregulate SLC6A1 at each of those points to improve the disease condition? And how much upregulation is too much? When does it become toxic? Which we haven't seen yet, which is good, because a lot of these diseases, too much function causes problems as well as not enough. Okay, so it was a brief talk. Here's our SLC6A1 team at the Flory. There's also a couple of parents in here, Stuart and Vicky Place, and their son Will, Will's got a frame shift mutation in SLC6A1. Emma's son Thomas isn't in the picture. He was being naughty that day and couldn't come. But this all kicked off in the first instance through a donation from Emma, and Stu has been instrumental in securing funding to allow us to do this work. Stu and Emma and Vicky have started a foundation called SLC6A1 Kids in Australia, which is very new, nothing like SLC6A1 Connect, but expect to hear something in the future. Everyone's contributed a lot to this work, but honourable mentions are probably, there's Mel sitting there. Snez is also in the photo. Snez is a group leader from the Flory. And on the very end there is Jana, second from the end, who has done most of the in vitro screening for this work. So does anyone have any questions, except for what the mechanism of action and the targets are? No? Yeah? We're using a couple. I think these are KN60. We've got a few different neuroblastoma and glioblastoma cell lines. I think off the top of my head these are KN60 and what is AN68 or? We started in HEC, but the expression wasn't high enough. Off the top of my head, I can't tell you the other glioblastoma cell line that we have, what the initialism for it is, but we're sort of using that as a backup because they're semi-suspension cultures and it's very difficult to do high throughput screening in those, whereas the KN60s are adherent monolayers. Yeah, sure. Sorry,
Yeah, so my question is, you mentioned that you use a group, but rather you remove the group, so you can increase the
yes.
expression. But is the group specific to SLC?
Yeah, so in the in silico design of the ASOs, we look for off targets, and so by an off target I mean that anything else in the genome that ASO could bind to, and we exclude those. And as part of the screening, we also hit NGN2, so iPSC-derived neurons and immortalised cell lines with the ASOs, and look for changes in expression of anything that isn't the target and exclude those ASOs as well, just to get around accidentally up or down regulating something that we're not aiming for. So yes, they are specific for SLC6A1.
Have you had success in similar types of disorders?
We have 3 that are very close to going into non-human primates. So this is the pipeline that we've developed. This is the pipeline that we've developed with those programs, and those are in conjunction with Praxis. So we spent a lot of time and money to get those right. And now we've set this up as a platform where we can just try and punch through to get ASOs out at the other side quickly and relatively inexpensively. So that's with the whole IP thing, is we're looking to protect the IP so that it's a valuable package for someone, whether that be Praxis or someone else, to take over financing of that. Because once it gets to even non-GLP tox in non-human primates, is about $3 million US a go. So the further you go, the more and more expensive it becomes. I think that, yes, yeah.
Under all circumstances, when might this therapy become available?
This is why my boss would have been better to give this talk. So he's been involved in a number of first-in-human trials of ASOs, and Praxis have some underway at the moment. We would expect this would be in a few years' time in the case of ASOs that work for the bulk of people who've got haploinsufficiency in SLC6A1, and they would have gone through GLP tox trials in non-human primates and probably efficacy, because we try and, where we can, we try and make the ASOs non-human primate human cross-reactive. But for N of 1, I would say that probably within 2 years, it's possible to have a safe, potent ASO. Certainly that's one of the plans we have for this.
I have a question here.
Yeah.
So thanks very much for the talk. So ASO is known also to downregulate mRNA in some design. So it's very powerful that you can upregulate. So I'm wondering, especially related to Kenny Kang's work, that the variant of the SLC6A1 might stuck in the ER. So I'm wondering what is your thought on using ASO to downregulate that expression of the variant?
That's a separate project that we have that we're applying for funding for at the moment, and that would be allele-specific ASOs to try and target dominant negative alleles. I don't think that there's any good evidence in the literature that there is a dominant negative effect of SLC6A1. Certainly the cryo-EM structures seem all to be monomeric, and all of the machinery for GABA and ion transport is contained in one subunit. So I can't rule out that there is a dominant negative effect, but it becomes a lot more complicated. And then what you're looking at is either the SNP that causes the mutation, you have to target that, which is very difficult, or you would be looking at a case-by-case basis where you look for SNPs with a higher minor allele frequency that are present in the same allele that the SNP that causes the mutation, that causes the disease mutation is, and targeting those SNPs, it just becomes a lot more complicated. But certainly what you're talking about is gapmer ASOs, where you target the mRNA for degradation, and that's not entirely what we're doing here. But without giving too much away, if there is an mRNA species that causes downregulation of your primary mRNA transcript, you can target that with a gapmer and, like I say, release the brakes from expression of the primary transcript. Does that make sense? Any more questions? Okay, I'll leave it there.